Welcome to the Center of Biotechnology and Bioengineering. I said it wrong. Center of Bioengineering and Biotechnology's very special event. We also call the center CBB because it's quite a long name. And this is our second Bioinnovation seminar in which we bring world experts to talk to us. I'm Shirley Fenton, and I'm the Business Development Director for the center. Normally, our director, Dr. Catherine Burns, would have this pleasure. However, she has other demands today. One of her PhD students is trying to defend the defense of the thesis today, and of course, she has to be there. We're delighted that you have joined us today, but before we begin, I'd like to recognize GE Healthcare, who has graciously sponsored financially this special event. With us today, we have Barbara Leone, which is back there, and there is a table of materials for you to take a look at out in the foyer. And as well, we have with us Fiona Fitzgerald. So Fiona, would you like to say a few words, please?
I certainly would. Thank you very much, Shirley. GE has been involved with the area of regenerative medicine now for 5 years. And we, in fact, we just have started to open our new Engage Centre. And this is a centre that will be involved with development and biomanufacturing, so scaling up and scaling out in regenerative medicine. It's based in the Mars building downtown, and we are currently recruiting, and we will be recruiting for the next while. So please do consider us when it comes time to send out those resumes. So regenerative medicine has been a big focus. We have some of the answers around the regenerative medicine scale up and scale out tools, but we don't have all of them at this point. And it's a field which is emerging. It's a field where there's a lot of research and there's a lot of business potential in terms of commercialization as well. So this is why we have we're really pleased to sponsor this this talk today, and welcome you very much to here. Thank you very much.
Thank you, Fiona. Yes, thank you. Next, I'd like to introduce to you Dr. John Thompson. He is the Associate Vice President of Research at the University of Waterloo, and he will introduce our speaker. So, John, if you want to go ahead.
Okay, oh, I need this. Can you hear me? Yes, this is working now. Thank you, Shirley. Thank you very much indeed. And ladies and gentlemen, it is a singular pleasure for me to introduce our speaker today, Professor Zhanfeng Cui. Professor Cui's career can only be described as distinguished. He is currently the Donald Pollock Professor in Chemical Engineering at the University of Oxford in the United Kingdom. And he is also the director of a very important center. It's called the CRMI Technology Center at Oxford, and it's a center sponsored by China Regenerative Medicine International, which is a public company in Hong Kong. He received a BSc degree from Inner Mongolia Polytechnic University in China, and MSc and PhD degrees from Dalian University of Technology. Following a postdoctoral appointment at Strathclyde University, Dr. Cui became a lecturer at the University of Edinburgh in 1991, and then moved to Oxford in 1994 to take up a position as university lecturer. In the year 2000, he was named the Sir Donald Pollock Professor of Chemical Engineering at Oxford. And for those of you who don't have first-hand knowledge of Sir Donald Pollock, and I confess to being one of those, Donald Pollock served as a commander during the Second World War. He was also rector at the University of Edinburgh for a period of time, and during the course of his long and distinguished career was chairman and president of several large companies. Sir Donald bequeathed funds to endorse a readership in the Department of Engineering Science at Oxford. And in 2000, in the year 2000, these funds were used to establish the prestigious Sir Donald Pollock Chemical Engineering Research Chair, the position that Professor Cui now holds. It bears noting as well that Professor Cui was elected as a Fellow of the Academy of Engineering, the Royal Academy of Engineering, sorry, in 2013. His main research interest is in developing enabling technologies for regenerative medicine. And the center that he now directs conducts translational research targeted to many of the debilitating diseases that continue to afflict humankind, including cancer, diabetes, neurodegeneration, and musculoskeletal afflictions. Among his many research accomplishments, and I was very impressed by this because I've done tissue culture myself and it's not easy. Among his many research accomplishments is engineering a three-dimensional human neural network, which involves culturing stem cells and enabling controlled differentiation of these stem cells within a three-dimensional architecture, directing growth of the neurons using concentration gradients, cell positioning of the neurons within the network using surface chemistry, and of course data analysis using very complex modeling techniques. And that sounds very, very difficult. I must admit it does. Dr. Cui describes research as being fun. He also teaches at Oxford and describes teaching as being enjoyable. And here's the funny part. He also dabbles in administration, which he refers to, and here he is somewhat equivocal, unlike his positions on teaching and research. He describes being involved in administration as, well, being, well, good training, sort of in that equivocal context. And yet the funny part is, I sense from that administration is not a priority for Dr. Cui, but the administration, the senior administration at Oxford, they've identified him as someone who can do administration very well. And so he gets more and more of this administrivia piled on him. And I sense, sir, that you have difficulty saying no to some of these senior administrators at Oxford. Anyway, you do all three, and all power to you. That's really commendable, given your involvement in research. So the title of his presentation today is Regenerative Medical Technology, from Fundamental Research to Commercial Biomanufacture. Zhanfeng, we're delighted that you've been able to visit us. We do look forward to hearing your presentation. And friends and colleagues, please join me once again in welcoming Zhanfeng Cui to Water.
Thank you, John. Well, good afternoon, ladies and gentlemen. And it's a pleasure to be here. And thank the university, CBB, Shirley, invite me to be here. And you just listened to what GE Healthcare, the lady from GE Healthcare, and just said, and John said, actually that was the message I want to pass to you. And then we can finish this seminar, actually. That's the message I just want to say. It is a pleasure. This is my first time come to Waterloo. Of course, I heard a lot about this place. And so it is also, I'm very pleased to see university have this CBB, Center for Bioengineering and Biotechnology, and as actually umbrella to encourage all the collaborations between different departments. Because I do believe nowadays the problem we're facing now really cannot be solved by a single discipline. If they can, they're already done. So what's left for us is all the difficult ones, and usually it's very difficult for one discipline to solve the problem. So this kind of multidisciplinary approach particularly utilize advances in biotechnology and the bioscience is actually they open a lot of new doors for us. I think that's what I see a great opportunity there. I come from University of Oxford, and the people quite often say, I say Oxford University. They would say, is that the University of Oxford? because I realized in America, it could be different. You put the first word or second word. You have a University of Washington or Washington University, they are totally different. I said the Oxford is only have one, so it's all the same. Now, I come from, I do a little bit of publicity for my employer, very famous for non-English speaking word, because we got a dictionary. It's probably the most famous one. Also made probably more money for the university. And we produce a lot of tech politicians. And even the current one is also all graduate from Oxford University. And we have very good architecture. And the people come to Oxford, admire or study architecture. We still have a lot of strange systems. You go to a dinner, you have to wear a gown. You go to exam, have to all dressed up. You see, for all the formal exams, we have to dress up, so-called sub-fast, you see. Otherwise, you cannot go to the examination. So we have famous people. This is a guy just got a Sterling Prize. What is the computing people? This is Tim Berners-Lee, got the World Wide Web. You see, the inventor of www, internet. And so now we just made him also professor of Oxford Computing Science. He's our graduate. And then we have Roland Atkinson. And he's a graduate from my department, engineering graduate. You see, the Mr. Bean, right? So we, for some reason, last year we are ranked the #1. So we still don't understand why we ranked the #1. But anyway, we said that must be a good thing. So Oxford is full of traditions because it's 860, 70 years old. And so I saw the university here is, the country, Canada, is 150 years, right? So then this is much older. And so we have a combination of old and the new. So this is Christchurch, one of the oldest colleges. And so this is where a lot of Harry Potter film was shot there. So now children come to Oxford, this is a must see. And then we have some new colleges, like this is St. Hugh. This is a women's college, started in the 1890s. Then this is some old very thing, and this is 1960s. Wolfson is a new graduate college. So we have 39 these colleges. So when we say college in Oxford, it means totally different things. It's like a little university. It's not a college of engineering, college of science. It's an independent, financially independent setup. So Oxford University is one university plus 39 this type of small universities. But it's the same group of students, the same group of faculties. So this is my department that we have called it engineering science. We put everything together. So we don't have a subdivision. So we have mechanical, civil, electrical. That has been started in 1908, just over 100 years old. And 1987, we started information engineering. 1993, we started the chemical engineering. About 2000, we started biomedical. So now the student come to our course, undergraduate, the first 2 years all exactly same. And then the third year, they start to become specialized. So they opt the final year, 4-year course, they opt one of these options. We have about 100 faculties, and we are in 2014, again, for some reason, we even beat Cambridge University in UK. We become #1 in the UK. And then we are #3 in Europe, in the world, the #1 in Europe. This is in the last rating. I think sometimes we assume our vice chancellor or president must have did something to get us to the top chart. Anyway, so what do we do? This is CRMI Center, my research. This is actually, this CRMI is a public company in Hong Kong. They list in Hong Kong. And then later they find that we can do something to help them. I will tell the story later. Basically, they give us £9 million and for 6 years. Because at the moment in the UK, research funding is very short, very difficult, and particularly the Brexit now. And so we are in the second year now, so this money help us, my center, pass the difficult times. So that is very useful. So we're working on enabling technology related to stem cells and the tissue engineering, doing work on biomaterials. Then the applications, we mainly focused on diabetes, cancer, neurodegeneration, and musculoskeletal science. So repairs. Now, I think I put a couple of slides talking about the regenerative medicine. So what are they? It's really trying to treat a non-curable disease and conditions. I put the non-curable there as actually a very important part because this is clinical need. If somebody take a pill, solve the problem, why you bother with stem cells? Right? So basically, when you get the non-curable, we can't do anything, like degeneration, like tissue organ loss, and now cancer. And then this regenerative medicine offer a new approach. So then either use stem cells or stem cell and cell therapy, and the tissue engineering, and the use of functional molecules. That's the general approach. Or you can put them all together. And so this is just a couple of pictures from a website. So the idea of tissue engineering, original idea, in 1990s, is just to put, if we need some tissue, and then a piece of bone, for example, you can use imaging. Based on the imaging, you can do prototyping, and then you can get a scaffold. And put the cells, put some goodies, growth factors, keep them alive. And in the lab or in industry environment, you can grow into a piece of tissue, then you can implant. So the idea is very simple. And then, actually, 1999, the first tissue-engineered live skin products was launched in the US. So that's the first product. And for stem cell therapy, it's just like people often use this example, but whether it works or not is still on doubt. So if you got this patient got Alzheimer's, and then a lot of tissue loss in the brain, so all the connections are gone. So no matter what drug you take, they can't do the repair. So if you feel that with stem cells, and maybe stem cells can develop into neurons and can make the connections, and then you get prevent further memory loss or try to get function back. So that is actually the simplified idea. So whether it works or not is still debate. And the question, and what is the issue is that the more research we do, the more complex it looks like, and the more difficult to solve the problem. Right. So, right. So this is the first tissue engineering products for skin. And for stem cell therapy, and then there are two approaches, right? One is like ideal scenario is stem cell medicine, okay? You just watch like the Terminator film when they lose all the tissues, you inject some cells, they grow, repair, that's great. One product fit all, everybody can use it. But the problem is the immunorejection, tumorigenesis. So as all this still, so far, there's no products launched in that area. So people think probably the most likely one will be mesenchymal stem cells, universal mesenchymal stem cells, but it's still sometimes long way to go. So what can be done now is the autologous stem cell therapy. Basically, use patient's own cells, you do a processing, and then put it back. But then, so that's you don't have this tumorigenesis issue because it's all adult stem cell, and then it's own cell, so you don't have immunorejection. So this is a low-hanging fruit. So at the moment when you say stem cell therapy, clinical application, almost sure is use patient own cell, apart from bone marrow transplant, which use cord blood stem cell. And so this can be done now, but the main problem is cost. So for treatment, if you cost $50,000, so it only benefits a few rich people, then probably it's not a good idea. And then, so this is, the other thing is cannot provide off-the-shelf availability. When you need it, you have to take cells from patient, take some weeks to get the cells ready for put back. So this, but then, so there's some examples. You probably read all this, see these stem cell hypes. So get a paralyzed patient and get it back. The technology developed in London, in UCL, but the British system is very difficult to get clinic. So this professor went to Poland to treat this patient and get it stand up and walk. You know? And so then you can have to treat diabetes and the cancer. A lot of this related in this line. So a lot of papers come out. But if you're looking at, are they successful? And so its stem cells is still much newer, and then you're looking at this tissue engineering. This idea, when Bob Langer proposed this in 1990s, and now it's 20 years, and then you say it's a success. But what on the market is 5 products on the market. It's all skin products. There's no blood vessels because we'll try to heart bypass, and there's no live bone products. There even no, you see, people initially promised so much, but now only 5 skin products. So then a lot of the applications you see is the tissue reconstruction. So for individual, when this patient, they need a bladder. Oh, let's make a bladder for this patient. But it's not a commercial production. Okay, it's all individual case. And then quite a lot now used in plastic surgery. You see, I want a bigger nose. I want to change my ear. So that is actually used a lot now, and somebody make a lot of money, right? But then it's not really clinical need. That is where we should focus. So then I said, why is that? Because basically we don't understand it. Okay, it's a lack of understanding of fundamental science, even how to grow cells, how to control the stem cells, and then it's all these fundamental sciences. And then I think it's a misunderstand the clinical need. Because a lot of people working on universities, I said, I just want to publish good papers. All right? And then actually what we produced, what we delivered, is not what the surgeon wants. So actually quite often that is the issue. In one of the bigger conferences, and this person gave actually a presentation about IVD, in vitro disc, right? Intervertebral disc. This is where you got a lot of people get back pain. This is my tissue engineer. The disc looks like this, tested like a disc. And then a surgeon standing up said, how I can put this disc into the patient? And this presenter said, that's not my problem. All right? So that is the issue, you see. And then the knowledge of biomanufacture. This is, you see in biochemical engineers, after this 50, 60 years development, we know how to make monoclonal antibodies, we know how to make vaccines, but now how we make tissue and life products. Okay? So we don't know. And then the regulatory framework, and is actually quite, a lot of people, we don't understand it. So we do research, and then when later you said, I want to commercialize it, how can get the FDA approval? I said, you can't, because your starting point is wrong. So that is all these issues. So that's, I wouldn't say this is a big success. So this is related actually to the manufacture. This is the first product, the Appligraft. You see, that's outside Boston, the Organogenesis make it. It's quite complicated, but what I want to just point out, the timeline. So this is day 0, day 6 you do something, day 10 do something, day 21, 20 days. So to grow a piece of skin here, take 20 days. Now, in chemical reaction, we want reaction time as second or minutes. Okay, you can wait for a little bit longer. If you make a yogurt, you leave it overnight. Okay, now to make the skin, you need 20 days. Anything can go wrong within 20 days. So that's, and how you make a lot, that's another thing. So this is, first thing you have to make it, then you have to make a lot of them and make uniform quality. And the important thing, and now become more important, is affordable. Because in the UK, certainly, and in the US, every country, now the affordability become an issue. You can have a products, but not many people can afford it. And for stem cells, the same. So stem cells, we know stem cells can grow themselves, replicate, and then can differentiate, can change to the cells you like. And then, but then how you control them? You know there's a lot of factors, but then how you first thing we have to understand how to control that. Second thing is how to create the local environment to make them differentiate into the stem cell, into the cells you like. So a lot of them, we don't know. That's the problem. So now people say, okay, embryonic stem cells is great. You can treat a lot of diseases because potentially they can grow into all different sets of cells, different kinds of cells. But if you don't purify the products, if you have some embryonic stem cells left in the products injected body, and there's a high chance they form tumors. So because all the experimental data showed if you put embryonic stem cells into the body, 75% of chances will form tumors. So you cure one condition, you introduce a cancer. So that is, you see, it's not on. So it's all these issues. It's quite complicated. So this is the difference between science and engineering. Scientists can do 10 experiments, 9 failed, 1 succeeded, and maybe only 5% of cells turned into what you want. You can write a paper in Nature and Science. But then when you actually apply it to apply, you can't apply that. In industry production, you have to be, every time is right, is all successful. And then you to get the right quality. A lot of fundamental questions. So how to grow cells? I said, I said, we have a lot of cell biologists, I'm sure, microbiologists. I said, we know this is how we grow cells. My supervisor told me this is how we grow cells. My supervisor is right in this one. So, but then if you're looking at what in the human body, and the cells all in 3-dimensional environment, and they are all under perfusion. So this is actually, you see, this is a liver, bones, even in skin. It's quite a complex structure. So we grow cells in the lab, monolayer. What's the relevance to this, to human body, to what's in the human body? And they say, okay, why we don't grow 3D in perfusion? Why we don't mimic what's happened in the body? So basically, the difficulty is that, as I said, my supervisor told me this is how we grow cells. I can buy 50p by 96 vial plate, and the microplate readers are all designed to take the plate. And then you see, it's all this, even the microscopes, you can take all this established protocols equipment. So if I grow cells in 3D and the perfusion, I don't have equipment. And even I publish something, I don't have anything to compare to. So the referee will ask me questions, I can't publish my papers. If you're working at university as a graduate, you want to publish papers. So this is all these issues. Then we look into this when we actually grow engineered tissue. We said, very simple question, how we grow, what's the condition we use to grow these cells? So we developed micro bioreactors, basically perfuse the 3-dimensional culture. And it seems a very simple idea to mimic what's happening in the body. And then suddenly said, there's no equipment. We have to develop our own equipment. So this is actually very first one. This is probably the 2001, around that time. We started to make these little devices just for our own use. See, we can put the cells in 3-dimensional here. This is 96 vials, exactly same size. We put the perfusion in and out. Then this is transparent. You still can use microscope to see what's going on. And when we did that, and we published some papers, and then a lot of people say, why can you give us some microreactors? And we said, university, that's not our job. So we set up a spin-off company and in 2009, and just commercialize this. So this is actually an example that you actually very, very simple thing. How you grow cells. And then you see, it's give you opened a new area of research. So this is actually, we commercialize it. So this is the products. And then it's launched in 2011. And the company is still there and still making this kind of working on equipment for drug discovery. And now this company focuses on hepatitis B drug development now. But this is an example. Then we got a little device, but then this is not enough. You said that if it's 3-dimensional culture, you actually couldn't see it in an ordinary microscope. Because if you are at the bottom of the dish, you can put a microscope, you see all the cells. Now in 3D, and then this is ordinary microscope will not work anymore. So we either use confocal or this multiphoton. So this is actually the see cells in 3D. It works, thanks for the computer. So this is actually this is mesenchymal stem cells in 3D environment. So and when I presented this thing, and I give tissue engineering papers probably 10 years ago, and some guy in the audience from Pfizer come in and said, why don't you use this for drug testing? I said, what is drug testing? I have no idea about the drug testing because we are mainly focused on tissue engineering. And he said, why don't you do some experiment for us? So we did some experiment just to test the drug candidates. And that's make us move into drug discovery, drug testing, screening area. And so that's, and this is again, this is because the macros, you can looking at the collagen scaffold cells and cell in collagen. So there's one dead cells there you can see. So it's basically this 3-dimensional environment. You have to you have to develop new tools, not just the culture, the assays, the analysis. It's all this fundamental research side. For example, this is following a single cell, how they grow. When you put it in, this is day 0, and then you got day 1, 2, 3, 4, 5. And you can see the cell spread, the cell shape change, because of course initially they are round, they don't like the environment, and that's they're not natural. And slowly they adapt, and finally you grow that in the real cell shape. So it's all this type of analysis. You see, imaging, analysis, develop all these different methods. It is important. So why we do this? You see, then we have to demonstrate, because initially when we say for tissue engineering, of course you have to do 3D. You have to provide the nutrient. That's why you need the perfusion. So then when we move into this drug discovery, the application is for drug discovery screening side. So this is look at the pH. If you got to perfuse the environment, the pH not change. But if you use conventional cell culture, of course you have fresh medium, and then you can see the color phenol red change, and then to a stage you have to change again. So all your pH fluctuate. But it's not just the pH. Any other chemicals concentration fluctuate. If you actually test the drug, initially you have one drug concentration, and during the time the drug concentration change. So later, when you want to look into your drug kinetics, and then how the drug concentration affect the functions, if everything change, then you will have a difficulty to link them. But if you do perfusion, you know exactly the condition, then you don't need to worry about it. You can quantify the link. A couple of examples, I'm not going into great detail. This is to test diabetic drug. Okay, so this is one drug, this is another one, GLP-1. Basically, the lower line is a conventional way of culture that, static and 2D culture. When you got a 3D perfused culture, we do see the drug response. Basically, this is, you have to take my words, because basically, this is like in vitro. Just like in the body, and you increase the concentration of glucose, and then the more insulin come out. When you wash them out, no more drug or no more, and then the insulin secretion become lower. And the same as this, basically the profile is like what happened in the body. So this is for test diabetic drug, and this is for cancer drug testing. So this is actually why you see a lot of companies is that we have a great cancer drug, very effective in the lab. When you actually go to preclinical, further go to clinical trial, many of them fail. So this is actually, we believe there's the really problem is actually animals are different from people. And the preclinical study is all based on the monolayer cell culture, not to write the right model. So this not to use the right model. So this is actually all the data have 4 bars, and this is DLD1, basically breast cancer model cell line. This is colorectal cell culture cell line, and the 4 bars. This is different drug concentration. Now this is a conventional lab-based 2D cell culture. Now, second one is the current so-called advanced spheroid. A lot of people use spheroid. And the third one is this 3-dimensional culture model. And the next one is for the cancer tissue slice. You see, this is how effective it is. If 100%, basically all the cells get killed. So if you if you look at this one, for example, then you see there's 95% efficient if you do monolayer cell culture, and then you spheroid of 80, then actually our model is about only 70%. Then you got cancer slice is about 70%. The message is that the 3-dimensional model is more close to the test results from a cancer slice. So this is actually, when you got 3D, really make a big difference. So a lot of cancer drugs synthesize the compounds, very effective to kill the cells in the lab. But when you go to the clinical testing, they are not effective. When we try to persuade the drug companies to use our model, and we find it's very difficult. Because if you are a drug discovery company, you really don't want to know this problem. When you get here, you said, my drug is 90% effective. Then what they want to do, they want to sell this to a bigger company. Then they don't want to know you have problems. So that is actually, when you go to translational, go to commercial side, a lot of unforeseen scenario comes in. It's very interesting. So we're working on use stem cells, use different scaffold. We try to differentiate them, build up this various in vitro models for drug testing. So then we actually now working with, we couldn't work with a drug discovery company because they don't want to know they have problem. So we're working on the buyers. They need to know. They want to buy it, then you find out it doesn't work. They spend a lot of money. So we're working with big pharmaceutical companies looking into this issue. Right. Now, I said I would say something about I'm running out of time already. Huh? Okay, so I'm trying to speed up. This is actually more how to build up a human brain model for test. I said, why you want a brain model? Because it seems the brain is the last frontier. We try to work out how brain work. So you can do functional imaging. So you see you're laughing, some area lighting up, you're crying, some other area. And then you can get the cells, or a cell culture, but what we really want to do is to see whether we can get a 3-dimensional neural network. The problem with neural network is that the mature neurons don't grow anymore. So we have to start with stem cells and then let them grow to build up neurons. Because if you got mature neuron, you can't get that, they don't grow anymore. So we have a project. This is finished about 2 years ago, a 5-year project. We started this about 7 or 8 years ago. We have Ollie Paulson in Cambridge. He was in Oxford when he got this grant. When we got the grant, he migrated to Cambridge, and then Mike Coleman from Austin University. Basically, we try to work out the 3D neural architect. This is a bottom-up approach. Now, the idea is that if we can have a 3-dimensional neural network, so this is not a computing-based neural network, you have input layer, output layer, in the middle they have connections. So if we can get this build up, we can see how the signal get in and out, and then if we got this model, we can try to study how the neural network learn, or how they be trained, and then later we can look in the degeneration and the possible repair. So the first question we have, human cells grow very, very slow. Neurons. For the mouse, you take 6 weeks or 3-4 weeks, you can get a mature neuron, you can do experiment. But the human, we take 4 months, and they're still not mature enough. But you think about the child develop, you see through 20 years to mature, so all the neural system is still adapting. So this is actually one difficulty we have. And another difficulty is actually the cells are very mixed. You control the cell population, because you got just a neuron doesn't work. You need to ostracize, and then you all this right population, and then there's a lot of experimental work, and then you have to make sure they're functional. And this is just to show the timeline. So 3 to 4 months, 4 to 5 months. So take quite a few months just to get the cells from stem cell. Initially we use embryonic stem cell, later we use iPS, and then to get mature neuron cells. So that's the first challenge. And then again, this is just after 100 days, you still, I can't tell the difference, but Ollie Paulson said that they are still not good enough, you still have to culture that to have them get them mature. And then we need what kind of scaffold in the middle. So at that time, a lot of porous solid scaffold, electrospin fibers, and finally we settled we will use hydrogel. And I know we have colleagues here interested in hydrogel. And then we said, when we grow the neurons in 2-dimensional, and they grow very happily. You see the imaging, very nice. But you put 3D, all the problem appeared. You see they don't spread, and they trapped inside, and they have lots of red dots. The red dots are dead cells. Because if you're 2D, the dead cells will detach. What you see is our live cells. But in 3D, if they are inside, trapped, the dead cells are still there. And so we work out, the postdoc worked very hard. Finally, they work out, it's become actually actually, when you get there, when you solve the problem, looking back, you say, oh, that's so simple. If you got the cells trapped inside, then of course, everywhere you got the polymers, the neurite couldn't go out. So what you really have to physically create space for them to grow out. Then that's why they said, let's fragment the hydrogel. Basically, the hydrogel become fragmented and then generate the space. So the cells will grow through the space. So this is now we got 3-dimensional from different level. You can see now the neurite, the green stuff is the neurite grow within the 3D. So this is within a little space. You can see all the neurite now in 3-dimensional space. They are connected. So we now know how to grow in the middle part. And again, you need a directional growth. We want them neura grow one direction. So we use nutrient gradient to control them, let them line up. So you can see this is looking better, is all the neurons grow this way, so we can control that. And then we needed to control the connections. We said, okay, one layer to another, how we control the connections. That is not allow them to grow randomly. So we use porous membrane. We said, okay, only here have a hole, then your neurite can only grow down. You cannot grow other places. Physical barrier. Then we have to work out what would be the right pore size. And so we started with isopore membrane, and then look into optimize how they grow in or grow out. Yeah. And then it's very smart, actually. And they find the right place to go through. We're still trying to work out all the physical properties. You see, this hydrophilicity, electrical charge, the edge, all this have effect. They don't lack the edge, sharp edge. So we said, okay, but as you pore, they all have this sharp edge. So now we have to make our own membrane. In the department, we have a professor with this advanced laser processing. You can punch in holes for any materials, glass, polymer, Martin Booth's group. So we can actually get all the different, we even get a square. We can have different one. We can have a different angle. You see, curvature as entrance. So we can control this neurite where to end up. Now we now sort of the middle part and how to grow the growth. But what about the input layer and output layer? So the input layer, we said we also need a microscope on the top to see. So this top layer has to be transparent. We can see inside. And then we need the cells sitting somewhere so they grow into it. So we borrowed the idea from Stanford, basically to introduce the top layer of cells, introduce a gene, optical sensitive. You can optical activate. You share light, the cells will activate, get the signal out. So this is actually get its plasmid into the cells. This is the work done in Aston University. I'm an engineer, you see. And so this is actually, we work out the cells. They got infected with GMP labeled, so you know they got transfected. And then they do have the typical, they develop into neurons after the genetic modification. Then we can position them on the top. So now we can share light on the top to activate, to stimulate the cells. The cells get the signal, go to the underneath, go to the middle layer, and then from the middle layer and then go to the bottom. The bottom layer we use microelectrode array. So this is 256 dots, and then you just hope some of cells in theory, we can position them, but in practice, just random, we just hope some of cells will sitting at the electrode. So we can pick up the signals. This is actually pick up signals. So then we can all this combined together. You have positioning, control the growth, directional growth, three-dimensional culture, because you have to supply nutrient to the middle part. And all this come together, and we can get signal out. So this is, now we just need to share light. And then you can just get light activated at cells. The cells will generate a signal as it go to the bottom, and then this electrode will pick up the signal. So we generate a lot of data. Now become a difficult part. You see how you analyze it. And then then this is actually the more challenging part. So we haven't published the data yet, and because there's a lot of data sitting there. But that's, at least now we can get this built up. It's great fun. It's 5 years, a lot of frustration, and spend a lot of money, a lot of people involved. I say overall about 20 people involved. And the Professor Ye is also a co-PI on this project. So we said, what's this used for? Well, I think this is another, for fundamental research, let's don't worry about when or whether it can be used. But then for the sake of argument, we said we got a model. We can actually do drug testing. We said if you try alcohol, where alcohol affect the neural development, for example. And so it's for stem cell therapy for safety and efficacy assessment, because neurodegeneration is the main target, one of the main target for stem cell therapy. So as a safe, this type of thing, we need to worry about that. And then it's basically research too. This is actually a steep learning curve to us, and we know nothing about the neural system when we started this, and we said, but we know we only want to do human. We don't want to do mouse or rats, because we realize the human are very different. I actually spend a few minutes about this biomanufacture part. This is another interesting scenario, and this is written in Chinese because this company is in Hong Kong. And so this is the world's first tissue engineering the cornea. Next one would have English word. So this is a BBC made a program. BBC made a program of the 5 great science advances in China. And then this is one of them. So this is BBC's program. They said in China, because people do not donate, so there's a big shortage on cornea. So if people need a cornea, they have to all smuggled from India or Pakistan or Bangladesh. And so this company, CRMI, and through maybe 7 years of research, and basically developed a process, use pig eyes, pig cornea, and decellularize it, and then find and get rid of the cells, DNA materials, and the antigens, and finally left this transparent collagen scaffold, and directly for transplant. Okay, so that is the idea. And so this is the manufacture process. And you have a lot of pre-treatment, cleaning, sterilization, preparation, because the eyeball of the pig must be taken out immediately after the pig is killed, because otherwise they will denature, and then they become not transparent anymore. And then it has to be in under 4 degrees, moving to the plant, and then cut the cartilage out, and then you go through a process of decellularization. This is a 60 hours operation, 60 hours, and every 2 hours, every 1 or 2 hours, you change the solution. And so it's quite labor intensive, and each step is, you see, quite manually done. And then you have post-processing, all this quality control stuff. So what they have been doing is use the shaking bottles. Okay? So they got the shaking bottles. They said, right, we got the products, so they are submitted to FDA, Chinese FDA approval. And they know they will get it. All the feedback is that we'll get it. And suddenly realized how we make this. How we make this. And so you can't use the bottles. Each bottle you can put 30 maximum. And then they want to make 300,000 per year. They said, we don't know how to make this. So they actually come to me, because my student knows somebody there. And they said, that, can you help us? We said, we don't know. Nobody knows how to make it, right? So we said, we give it a try. So it took us about 8 months to get this to automated system, everything automatic system. And then we include the quality control. And then we got this assembled in Oxford, shipped to China, because they plant in China. And so they said, this is following this. They said, you can do some useful thing. So why we actually collaborate? So that's why they give us £9 million for 6 years. So they know we can do something useful to them. I think you see the industry people like GE Healthcare, you are smart people. You're only when you are sure that you can do something. Anyway, so this is actually the BBC News. If I can, maybe I can do that. No. there is a sound. But this is a BBC program, basically showed this, it's really a lousy title, Pig's Eye See the World or something. So it's really not. I think the sound is gone. So what, basically they just show a case study, BBC program. So you see, this guy got the cornea damage through work, and then you got the implant of this cornea, and then he can see it. He just got recovered. I forgot to ask to connect the sound. Anyway, so we can, basically it's just a BBC program, and then it's just like, I was interviewed by BBC, said that they asked me to say about this, to interview, live interview. I said, the first thing I want to correct you, this is not a pig cornea. You can't say implant a pig cornea to a patient, you see. Anyway, so this is actually, I will just get this off. So it's actually, he showed is recovered. So I want to put this into I want to put this story to complete this talk. This is a penicillin story. You said probably the single drug saved the most lives in the world is penicillin, right? And then you look at the penicillin. This is a discovery. You see, Alexander Fleming discovered accidentally penicillin. He got a Nobel Prize. And in the 40s, the three guys in Oxford, the Down School of Pathology, they worked out how to produce a single dose or a few doses of penicillin. And they got the Nobel Prize. And later, during the war, Merck got the contract, started this, forced engineers working with microbiologists, and work out, and also by luck, they work out how to produce large-scale production of penicillin. And then, under the war, thousands of people saved. Now penicillin is just like a bulk chemicals, right? So if you put this here, you said, where are the stem cells, where we are now? So we probably just are here, right? So this is why you see manufacturing issue, we don't know how to do it, distribution, delivery, all these things. So, but one day, stem cells, we will like a stem cell medicine. We just, everybody can, one product fit all. Now, this is actually, you said that this is still a long way to go, where you can make money. For those, I know this university, very interested in spin-out companies. So this is where people make money. Supply chains, okay? And also, you see service industry. A lot of CROs and doing that. And then I mentioned this in vitro drug testing. And that is because it's a lot of, we try to, if you can improve the outcome, the accuracy of the prediction, then you can potentially save a lot of money. So that's where it's make money. So I think the future is bright because the unmet clinical need, they are real. Because there's no alternative. And this regenerative medicine could possibly offer alternative solution. That's why I said the future is bright. But they really need people. One of them need a lot of money, a lot of people, interdisciplinary people, people with open mind to work together. And then I find I'm very easy to find the clinical collaborators because doctors, they have a real problem. They said, I want to solve the problem. What I find very difficult to find a good collaborator in cell biology because this top scientist said, I'm only interested in the marker of this pathway. And then I have no interest in that because I just want to work this out to get the Nobel Prize. So that's, we need the people. So I have been very lucky to have a lot of great students and postdoc, young, ambitious people working for me. I'm very grateful to them. And then I like to thank people giving money, people did the work. I'm just the one standing up to give this talk. Thank you.
What a fascinating presentation. Thank you very, very much indeed. Okay, so this presentation is now open for questions and comments, folks. Yes, please.
Just wait. I like how you take until we get that.
I like how you take on the biomanufacturer. But the last example you showed, the cornea, it still doesn't have cells, right? It's decellularized in a sense that it's not alive anymore. So to produce something with cell, even as an in vitro model that you can ship into research labs, that's still difficult, right? Like, do you foresee your 3D neuron network can be shipped out one day to even research lab who is doing tissue engineering? And basically, we can take that and just move on with it?
The answer is yes, because actually we work on the whole supply chain area. One of the key issue is preservation. And then you use cryopreservation or a neural model case. Depends on how big the model, the in vitro model is, the physical size. If they are small enough, you can vitrify them, so you preserve the structure. And otherwise, you have to cryopreserve them. You have to control the ice crystal size, not to damage the neural connections. So the idea, the long-term plan, is you spend 5 months to grow them, and then you freeze them, and then when you need it, you just take them out and do the testing.
I see.
Long-term plan.
Okay. I have just one more question. You showed the comparison between spheroids and your 3D model, because spheroids nowadays is considered as a 3D tissue model as well, right? I wonder what's the major difference between yours and spheroids?
Okay, because spheroid is very easy to form, and many like cancer cells, liver cells, they naturally form that. But you cannot control the structure. The only thing you control is the size. And then, so for us, for the if you actually introduce a scaffold, and then you introduce a freedom of operation, you can actually change the structure, and so that's under different cell distribution, for example. That's all can be done. And then the last couple of years, a lot of hype, let's put interest on organelles. Basically, is between the spheroid and some kind of structured stuff, and with multicellular cell type. But again, if you do testing for screening, you really want the reproducibility and the uniform. And then if you do this spheroid and the organelles, and it's very difficult to get all exactly the same. If you don't need to worry about the distribution, a little bit wider distribution, it's fine. But then it's just like use a scaffold, you can control if we call that quality, uniform and reproducibility. Yeah.
Other comments, questions? Well, maybe I could ask a question if I've got time. I have lots of questions anyway. So let me start with this one. You mentioned that one of the disease states that you're working on, Zhanfeng, is cancer. And I guess I see a disconnect between the cancer pathology and regenerative medicine. We didn't really talk about this. But a cancer cell is a cell that's targeted to die, yet doesn't die because of mutations in the death pathways, if you will, that regulate cell death. And so the objective is to kill cells. How do you do that by regeneration?
It's a good question. I think there are two aspects. So one is actually the stem cells and the cancer cells actually as a very thin dividing line, I would say. Because actually, this is another issue with actually the stem cell therapy. Because actually, if you grow cells, primary cells like a bone marrow, take some bone marrow cells, we want to grow them. Initially, they grow very slow. And after a few passages, they actually start to grow much quicker. And then when you're talking to the cancer cell biologist, they will say, that's no longer your cells anymore. Because all the P50 or your upgrade is all turned into cancer cells. Is how can you ensure they are still safe to use? That's one of the questions, one of the issues we have, somebody needs to address. Mm-hmm. And the second thing you see, to study cancer, particularly cancer stem cells, and the stem cell biology, they are so similar. They are so similar. I think to control that would be quite help each other. Yeah. But then what I say stem cell, what I say here for cancer cell therapy is not to use stem cell therapy. It's for related to grow a lot of cell therapy for now, for example, for CAR T cells. Now, what we envisage is that eventually we don't use patient own T cells to introduce a CAR, to introduce all the target recognition site. You can start, for example, use cord blood stem cells from our progenitor cells, and you can program them. You can grow a lot of them, and everybody can, people, or subset of the population can use that directly for cancer immunotherapy. It's just what we're working is to grow a lot of cells. Put it that way.
Okay, good, I understand.
Yeah.
Other comments, other questions? All right, at the risk of... oh, yes, please. Two up there.
Thank you for your talk. So you talked about drug testing on cells, right? For example, neurons is one model for drug testing. So recent developments are by using organoids, right, which are a miniaturized version of an organ. So what do you think about testing drugs on cells versus testing drugs on organoids, which probably represent, which are more anatomical structure of an organ?
Well, I think it's a bigger step moving away from just the cells. Because as I said, actually, I didn't give the data. For drug testing, for the clinical trial of drugs, 8 out of 9 failed clinical trial. One of the reasons is that I think the preclinical, basically they haven't got properly done preclinical study. So if initially it's all use cell lines, then now, for moving into organelles, because it's got the structure, got that, and even spheroid, we just answered that, is actually a big step towards the complexity. Or what we're looking for is a physiology relevance. So how they relevant to what happened in the body. So eventually, I would say there's a lot of interest to develop in vitro models. Initially, a lot of, say, lab on chip, this type of analysis. But then what I find, what we move away from that, we want to build up a sizable in vitro model to capture the three-dimensional environment. Because actually, if you do microfluidics, a little lab on chip, the surface to volume ratio is much higher. Then when you do drug testing, and then the results still you may not capture what's happening in the body. So, but then the question is that how close is close enough? And that is, you need a lot of research.
Other questions? Yes, here's one here.
Is there a big difference between the biomaterial that you use as a scaffold for 3D drug testing, like the hyaluronic, I guess, in one slide you mentioned, or between, I mean, different hydrogels or different?
The answer is yes, the scaffold play a very important role. But the problem is that I think it's not many people really want to spend a lot of time to look into scaffold. Because this is really, if you're looking at tissue engineering people, when looking at the scaffold, they don't really look into, okay, you want to grow this piece of tissue, what is the requirement to grow this tissue? So the first thing they see, what is approved by FDA? If we can test the materials can be used, they don't need to bother to look into the proper biomaterials. So I think it's really needed to get biomaterials people needed to, looking back, to say that if I want to grow this piece of tissue, this particular tissue or organ, so what is the best material? But you see, most people just go for the shortcut, said, okay, let's use this. And I think that is a bigger drawback. It's a material certainly is very important.
Thank you so much.
Anything else? Yes.
Thank you.
Thank you for your wonderful talk, Professor Cui. My question is, when you talk about you grow the cells, you use the isopore membrane. So my question is, why do you use isopore membrane? So is there any special consideration for the membrane material and the pore size? Thank you.
Yes. Well, thank you. It's a very good question. I criticize other people looking for shortcut. We are also looking for shortcut because we try to work out what would be the right membrane pore size for the neuron to grow. And then isopore membrane is a very low cell membrane porosity. Not many pores, you can see. But then one advantage is that they have relatively uniform pore size because the way they're made. So we can get commercially to get this membrane with different pore size. That's we evaluate the pore size. That's why we use the isopore. But it's not good for the application. So that's later we're moving away from that.
Okay. Thank you.
Well, thank you, Zhanfeng, for a very, very exciting presentation. I'm sure that if there are any students in the room who haven't yet made a career decision, your talk will be one that would certainly interest them, I think, in looking at regenerative medicine as a possible career, particularly a research career. I know if I were a young student again, I'd be perhaps knocking on your door. The other point I would make is that your talk is not only exciting, not only very interesting, and not only very compelling from the point of view of the story you tell and the progress you've made, but it's also, in my view, a very rich and compelling indication of the importance of interdisciplinarity. I mean, your presentation just rings with that. It strikes that chord very strongly. And so for all of us who work in science and do research, I think listening to our guest speaker today and reflecting on what he has said and seeing what he and his colleagues have accomplished should in itself be sufficient to indicate to all of us the importance of reaching out and collaborating with people in other disciplines. You've done that so well, sir, and we congratulate you for it. So, ladies and gentlemen, please join me in a very strong round of applause for Zhanfeng Cui. Thank you very much.
Thank you. Very kind. Thank you very much.
Very much.
Yeah, I hope that's okay. Can we take a picture?
Are you accepting students? Are there any people who want to sign up
come
today?
along. You see, because we have a few companies, and then if you want to join us after you graduate or come to do a PhD, you need if your VP write a good reference, I would take it.
Okay, so we have a small token of our appreciation that we'd like to present to you.
Oh, it's very kind.
Zhanfeng. I have no idea what it is. I'm not sure whether I should advise you to open it in our presence or not, but in any case, I do want to present this to you, and I think it would be, notwithstanding what I said, I think it would be very nice if you did open it.
Thank you.
Okay.
Right. I hope it's got the University of Waterloo sign on it.
But it's significantly appropriate, I think.
It's a flask-growing cell.
Oh, right. Indeed. It's got the University of Waterloo. Look at that. Well, thank you very much. Thank you, Shelley. Thank you, Shelley. Thank you. Yeah.
So I'd like to bring this to a close, but before I do, I have a special announcement. Dr. Cui didn't come by himself. He came with one of his colleagues, and she is giving a presentation, a lecture tomorrow, Chemical Engineering Department seminar, regenerative medicine from an engineer's perspective. So I'm sure that they would welcome you to attend that seminar as well. It's at 10:30 in EC6. I don't know the room, but I'm sure you can find your way there. Next, I'd like to just thank the CBB team, Christina and Chikima. We can't do these seminars without them. I'd like to thank the audience for coming. You've been great, very attentive, and your questions have been wonderful. And last but not least, the most important part is that we now have some refreshments and food, and we'd love you to stay and enjoy and continue the discussion. So thank you very much.